written in code. 4 letters, A, T, C, and G. These are the nucleotides that make up the material for our essential being, DNA. Humans are made up of thousands of genes, the coding segments that carry instructions for specific traits and proteins. But things don't always go as planned. Mutations occur when these essential instructions have a typo or an error, and the smallest typo can mean the difference between life and death. Recent research has revealed the potential for gene editing through a tool known as CRISPR. One night, 2 students are staying late to finish their biology lab project. Their goal is to successfully edit a gene and observe the effects. But one night, they find that their edited cells are behaving strangely. As the experiment spirals out of control, they must confront the truth.
Alright, we're editing the E. coli genome using CRISPR-Cas9. Target lac gene. Should be an easy fix.
What's CRISPR again?
Think of CRISPR as 2 parts, a GPS and a pair of scissors. The program follows instructions on where the gene is located and cuts at that exact place. It's not always perfect, but it's pretty cool.
That is pretty cool. I think our Cas9 protein's ready.
Me too.
Cas9 is a protein taken from bacteria and combined with a molecular scalpel that can cut DNA anywhere we tell it to. It's guided to the spot by a sequence of guide RNA that matches the target sequence of DNA.
Is the guide RNA programmed to find the sequence?
Guide RNA is programmed. Confirm target for the lac-Z gene, the one that breaks down lactose, the sugar in milk, into usable energy.
Imagine if we could do this for humans. We could edit genetic mutations that cause diseases like sickle cell anemia. It looks good. I think we can call it a night.
Me too. We'll check on this tomorrow.
Did you change the incubator settings?
No, but the colonies look mutated.
CRISPR is precise, but it's not perfect. Sometimes it edits the wrong place, and scientists call this off-target mutation.
I thought we were just editing a harmless gene.
You were, but if CRISPR cut somewhere else, you might have accidentally rewritten something vital.
Different editing can have different effects. CRISPR is used to add or delete a base sequence, but in this case, deleting the wrong sequence can lead to a frame shift, which completely changes the protein and the DNA itself.
The DNA is replicating itself?
No, it's not replicating itself. It's repairing itself using your new sequence as a template. It's rewriting everything.
In real cells, CRISPR relies on the organism's own repair systems. So if the repair goes wrong, the mutation can spread faster than anyone would expect.
We edited life itself.
No, that's impossible.
CRISPR doesn't distinguish between lab and life. Once you open the code, you can't always close it. That's why it's important to wear proper protective equipment in the lab.
I wanted to cure disease, not create it.
Breaking news. Local lab is shut down after a genetic contamination incident dealing with CRISPR gene editing.
Wouldn't it be dangerous to edit human embryos?
Yeah, it's called germline editing. And it changes the DNA of developing embryo for generations. Some say we're not ready for that.
That's why scientists debate where to draw the line. Somatic editing affects one person, while germline editing affects humanity itself. Even with that risk, CRISPR has shown the potential to cure blindness, sickle cell disease, and even cancer. But it's not magic. It's precision and responsibility.
The true horror isn't in the tool, it's in forgetting that the code of life was never meant to be rewritten without care, and our belief that perfection is ours to program.